Mendel's law of heredity. Johann Gregor Mendel was born to Anton and Rosine Mendel on July 22, 1822 in the Czech Republic. He was born into a family of Moravian peasants and proved to be very talented. During his childhood, Mendel worked as a gardener and studied beekeeping. Mendel demonstrated that the inheritance of certain traits in pea plants follows particular patterns, now referred to as the laws of heredity. Gregor Mendel is also known as the father of modern genetics. Gregor Mendel discovered the fundamental laws of inheritance while working on pea plants. He selected common garden pea plant, Pisum sativum, for the focus of his research, because it can be grown easily in large numbers in very less time. Pea plants have both male and female reproductive organs. As a result, they can either self-pollinate themselves or cross-pollinate with another plant. Apart from these, pea plants have strikingly distinct, easily detectable, contrasting features such as seed color, flower color, and height of plant. These are the reasons that Mendel chose the pea plant to conduct his experiments. He recognized the mathematical patterns of inheritance from one generation to the next. These experiments took him 8 years. During the experiments, Mendel grew over 10,000 pea plants, keeping track of progeny number and type. Mendel's work and his laws of inheritance were not appreciated in his lifetime. It was quite late in 1900. Results of his experiment were understood. Gregor Mendel studied 7 traits when doing his pea plant experiments: length of stem, tall or dwarf, color of unripe pods, green or yellow, shape of ripe pods, inflated or constricted, shape of ripe seed, smooth or wrinkled, color of seed, yellow or green, color of flower, purple or white, position of flowers, axial or terminal. He used several versions of these pea plants to discover the laws of heredity. Mendel's experiments. Mendel's experiments involved crossing and testing a large quantity of selected plants. His aim of doing these experiments was to examine how the plant's characteristics were passed on from one generation to the next. For 8 years, Mendel cultivated thousands of pea plants. Mendel conducted breeding experiment in 3 steps. Selection of pure parent plant. Production of first generation of plants by crossbreeding. Raising of second and subsequent generation by self-fertilization. In his experiments, Mendel was able to selectively cross-pollinate pure breeds having particular trait over many generations. In Mendel's experiments, when only one trait at a time was considered, the data was represented in monohybrid cross. The experiment dealing with a single character is called monohybrid cross. For crosses, considering only one trait to be analyzed, Mendel first selected pure line plants, means the plants that produced similar traits generation after generation. Pure line plants for height were selected. Tall plant on one side and dwarf plant on the other side. He then cross-pollinated such plants having the contrasting traits, considering one trait at a time, that is, height of the plants. These crossbred plants were either tall or short exclusively. In this experiment, the pollen grains from the flowers of a desired plant raised from the seeds of tall plant were transferred over the previously emasculated flower of a plant raised from the seed of a dwarf plant. After the transfer of pollen grains, the cross-pollinated flower was properly covered and seeds produced were allowed to germinate. Height of all the plants of F1 generation was carefully observed. Mendel found that all the plants of F1 generation were tall and there was no intermediate height. In this experiment, tallness completely masks dwarfness trait when true breeding plants are crossed. This shows tallness is dominant trait and dwarfness is recessive. Mendel then crossed the plants of F1 generation with each other. Means flowers of these F1 generation plants were self-pollinated. The flowers were kept covered from the beginning to avoid unwanted pollens from reaching these flowers. In F2 generation, Mendel observed the appearance of both tall and dwarf plants in 3:1 proportion. The breeding experiment dealing with a single character is called monohybrid cross. For example, when one trait like height of plant was considered, it was identified as monohybrid cross. Each allele is given a symbol. In the case of height, T might represent tall and T in lower case represents dwarfness. By convention, uppercase T represents the dominant, lowercase t represents the recessive. During monohybrid cross, Mendel found that offspring from first generation, expressed as F1 generation, always has dominant trait expressed in them like tallness in case of height of plants and purple color in case of color of flowers. However, the next generation expressed as F2 generation consistently has a 3:1 ratio of tall to dwarf. Mendel also studied the inheritance of two characters simultaneously. A breeding experiment dealing with two characters at the same time is called a dihybrid cross. Mendel considered shape as well as color of the seeds simultaneously. He crossed plants from seeds of round shape and yellow color with those with wrinkled seed and green color. It was observed that in F1 generation, all seeds had the features of only one parental type, that is round shape and yellow color. He raised plants from F1 generation seeds and allowed the flowers to self-pollinate to produce seeds of F2 generation. These flowers were kept covered from the beginning. In F2 generation, Mendel observed the appearance of 4 types of combinations. These included 2 parental types and 2 new combinations. Parental types are round-shaped and yellow color and wrinkled shape and green color. The 2 new combinations are round shape and green color, wrinkled shape and yellow color. The ratio of these phenotypes was 9:3:3:1. The breeding experiment dealing with 2 traits is called a dihybrid cross. For example, when 2 traits, shape of the seed and color of the seed, are considered, it is identified as dihybrid cross. Genotype symbols for round and yellow seed are uppercase R, lowercase r, uppercase Y and lowercase y. That is, heterozygous condition. 4 different gametes are possible and are produced in equal proportions. During dihybrid cross, Mendel found that offspring from first generation, expressed as F1 generation, always has dominant trait expressed in them, like yellow color and round shape. In next generation, means in F2 generation, 4 types of combinations were observed: round-shaped and yellow color, wrinkled shape and yellow color, round shape and green color, wrinkled shape and green color. These phenotypes appeared in the ratio of 9:3:3:1. Outcomes of monohybrid and dihybrid crosses done by Mendel. In a monohybrid cross, only one of the 2 contrasting traits appeared in F1 generation. However, in F2 generation, both the parental traits appeared in certain proportion. In dihybrid cross, when 2 contrasting pairs of traits were checked simultaneously, only one parental combination appeared in F1 generation. However, in F2 generation, raised by self-pollination, 4 combinations of traits appeared. These included 2 parental types and 2 new combinations. On the basis of outcomes of monohybrid and dihybrid crosses, Mendel coined several other terms and formulated certain laws. Before understanding these laws, let us understand how transfer of traits is controlled from one generation to another. There is a pair of factors controlling each character in pea plant, one inherited from each parent. Mendel considered these factors as a carrier of hereditary information from one generation to another. At the time of reproduction, when gametes are formed, these factors segregate so that each gamete receives only one factor of each character. This is called law of segregation. The law states that during the production of gametes, the two copies of each hereditary factor segregate so that offspring acquire one factor from each parent. Fertilization brings these two factors again together in the offspring. He crossed two heterozygous pea plants, which means that each plant had two different alleles at a particular genetic position. He discovered that the traits in the offspring of his crosses did not always match the traits in the parental plants. This meant that the pair of alleles encoding the traits in each parental plant had separated or segregated from one another during the formation of the reproductive cells. In F1 generation, only one character was expressed. Mendel called it as dominant character. The character which was not expressed was termed as recessive character. This phenomenon of appearance of only one of two contrasting traits in F1 generation is termed as dominance. This helps in stating the law of dominance, which says, in a cross between contrasting homozygous individuals, only one form of the trait will appear in the F1 generation. This trait is the dominant trait. At the same time, the character which does not get expressed in F1 generation is not lost. When F1 offspring were allowed to be self-pollinated, both the parental traits were expressed in definite proportion in F2 generation. From the F2 generation of a dihybrid cross, Mendel postulated that inheritance of factors controlling a particular trait in an organism is independent of the other. Mendel's law of independent assortment states that allele pairs separate independently during the formation of gametes. For example, the dihybrid RRYY produces gametes that have one allele of each gene. 4 different gametes are possible and will be produced in equal proportions. Means shape of seed is not associated with the color of the seed.